My name is Dave Vance, and welcome to a genealogist's overview of DNA testing science. This is a relatively quick overview. I'm not going to be going into a lot of technical detail about the tests and the technology behind them, nor about the genetics, to be honest. I'll give you a quick overview of both subjects, but there's a lot more to investigate, so if you're interested in following some of the detail, I'll give you some links at the end and in the description of the video so that you can follow up and learn as much as you feel like. This is an updated video from one I made about a year ago. I've incorporated both some very welcome additions and updates from Randy Harr, who went through the first video, provided me some suggestions, as well as I've got a little bit more in this video about some of the upcoming technologies that we should see as products within the next year or three, maybe, that will hopefully give us even more data than we have today. But I'm going to go through both the existing tests that we use in genetic genealogy today, as well as hopefully what's coming soon. Now, I'll start out by jumping straight to the punchline. Here are the types of tests that I'm going to be talking about today. So these are the current ones. I'll talk about the future ones in a second. Now, there are similarities under the covers of these. I tend to break it out into these because that's the major types of output that we get and the major types of genetic genealogy testing that we use in our field. So in the upper left, you've got autosomal tests are done through what's called microarrays, and I'll talk about each of these as a technology in a minute. So I'm just introducing them here, so I'm not going to go into a lot of detail. But this is autosomal, and frequently the mitochondrial DNA tests are done with microarray tests as well. You've got Sanger sequencing, which is the original. I'll talk about the history of these in a second, but this is really mostly these days used for single or panel STRs and SNPs. Not a lot of companies provide those anymore, but those that do, they tend to use Sanger sequencing for that type of a test. And then you've got next-gen sequencing, or what's often called short-read sequencing, which is another type of technology that's used today for big Y700, as well as whole genome sequencing and so on. And those use a different kind of technology that I'll talk about in a minute. Now, the upcoming ones, there are two. There's Nanopore from Oxford Nanopore Technologies, and there's SMRT, single molecule real-time. Also, it's been branded HiFi, and that's from a company called PacBio. And these are called long read. I'll get into what that means in a little bit, but these are future technologies. They're already being used very frequently in genetics studies, not so much in genetic genealogy yet. We've seen a couple of products in the past that used them or tried to. There's nothing currently as of May 2024 that I'm aware of that's on the market for genetic genealogy using these technologies, but they will be coming. They need to get some of the quality issues and some of the repeatability and the cost affordability of these down to a point that they can be offered as consumer products. So that's the overview of what I'll be talking about, and I'll get into some detail, again, not a ton, but I'll get into some detail about each of these. Now, if we stop for a minute to look at the history of DNA testing, what we call consumer DNA testing today really came out of a repeatable process that was developed in the 1970s called Sanger sequencing by a man named Fred Sanger. So he originally developed the first repeatable process that allows us to produce consistent DNA tests from human DNA today, or any other kind of DNA for that matter. But this chart you can see at the bottom where the source of it is a very useful chart to take a look at the history of DNA testing. Don't worry so much about a couple of points on this chart. Something called PCR was invented along the way, which stands for polymerase chain reaction. It's a method of amplification, and I'll mention amplification in passing, but I won't spend a lot of time on it. The other one that you don't need to really know for this overview is called capillary gel electrophoresis, which again is something that was invented along the way and helped improve the process. But what we call first generation DNA testing is really the Sanger sequencing as well as DNA microarrays. And again, we'll talk about those separately. Then along the way in the early 2000s, there was something called short read next generation sequencing that was developed, which also is called MPS for massively parallel sequencing. But this is what the basis for Big Y and the current whole genome sequencing is based on. And then in the early 2010s or around 2010, we started to get technologies that could do single molecule sequencing, and I'll talk about those in context as the future technologies that are coming down. And those are really what's called third generation sequencing. So we have 3 generations. It's important to realize that even today, though, the first generation testing has not been replaced by second generation. They coexist side by side and are very useful for different things, and we'll talk about that as well. So you get a sense of why certain testing technologies are used in some cases and not for others. So before we go into the testing itself, all the genetics that you need to know is on this chart. So you've seen the double helix that usually represents our DNA. Every rung of that ladder, if you will, is made up of what's called a base pair, which are 2 nucleotides. They're generally abbreviated A, T, G, or C. Each nucleotide is on a strand. The 2 strands are normally called the sense strand or the template strand, but you don't need to know that really. The key part is each of the nucleotides is a complement of each other. They bind to their complement. So C binds with G, T binds with A. If you know the nucleotides on one strand, then you know what it is on the other because they're just a complement of that one. So as you unravel the strands, if you separate them, you can read one, and usually you'll see it like I've written here as GTA, GAC, TAC, etc., one strand of these 4 letters. If you know what that strand is, you know the other one because it's just its complement, so you can translate them back and forth. So generally, we call them base pairs, but when we represent DNA, we represent them as one string of these letters because you can always recreate the other one. And that's pretty much all you need to know about DNA for this presentation. The other thing to know about DNA is that our chromosomes have been mapped, so these letters G, T, A, G, A, C, and so on appear in the same position in most of our DNA is mostly identical to each other, so we have the same nucleotides at the same positions. And those reference genomes, which is the map of where these appear, have been published and exist in the genetics world, and we use them in genetic genealogy during this testing to compare against and see where mutations have occurred in individual DNA. So all of us have GAT, AC, and so on here at a certain position in our chromosomes, and if you've had some mutations there, what we call SNPs, etc., are variations on that we can find by mapping our DNA with this reference genome against what we actually have in our DNA and seeing where the differences are. So that's a extremely high-level overview of that process, but we'll talk about how that works in some of the details of the different types of technologies in a second. One more quick point on this slide. If you do have a mutation anywhere along the sequence, let's say you have a T that changed to a G or whatever, that's usually called a SNP. It stands for single nucleotide polymorphism. Don't worry about that piece, but I'll be referring to SNPs in this presentation a lot. If you don't know what those mean, I would suggest you check that out because it's one of the basic mutations that we look for in genetic genealogy all the time. Now here, for reference, is a short glossary of terms that I may use or that you may see in context of any of these types of testing. They pretty much underline all of these types of testing in various ways, but they're terms that are used specifically in this situation that you may not see elsewhere, and so it's helpful, I think, to know what they mean, at least at a high level. Now, these are my definitions. They're not necessarily official definitions. But for instance, if I talk about denature, that really means separating the strands of DNA. In most of these types of testing, the DNA strands are fragmented into little pieces of various sizes, and that's a process that takes place as a step in the testing process, and then those little fragments are used in various ways depending on the type of test. For instance, binding or ligation means attaching pieces of DNA to each other, either synthetic DNA that you're attaching to a specific place on the actual tester's DNA, or sometimes you're combining other pieces of DNA together, or you can use a primer that's down below to attach DNA and so on, but that's usually called binding. Amplification, or PCR amplification, is when the fragments of DNA are duplicated. That creates more sample usually, so that you can work with more information, or work with more pieces of DNA to get the information you're looking for. A primer is a short piece of DNA that's created, usually synthetic DNA, that's created to match a section of the reference genome typically, and then that's because you're looking for a neighboring piece of DNA that you want to read and see if there's mutations there and so on, so you create a primer for it so that you can attach the other piece of DNA that you're looking for. Sequencing just means reading nucleotide values in series along any particular strand. Alignment is lining up reads usually after the DNA has been read, but it means lining up reads to recreate longer DNA strands. So the more you break it up, then you have to put it all back together. It's like a puzzle, and you recreate the tester's DNA by putting these little pieces back together. That is often easier if you have a reference genome that you can use as a guide. That's kind of like having the box top on your puzzle. If you don't have that, then putting the puzzle together is much harder. If you have the box top to look at, the picture, the box that the puzzle is supposed to represent, that's the reference genome in this case, that makes putting it all back together a lot easier. So that's all the introduction that we need to do. Let's finally get into the actual testing itself. So we'll start with the DNA microarray tests. These are typically done for autosomal or mitochondrial DNA. They're often called chip testing as well. And it starts with a chip. They build a chip of SNPs that they're looking for. They only test for known SNPs, so these are not discovery tests where they look for new mutations in your DNA. They're targeting anywhere from 600,000 to a million known SNPs that may or may not exist in your DNA. Now, bear in mind that we all have about 6.2 billion base pairs in our DNA that we could have SNPs on. So they're really only reading a very small fraction of that. That's why if you hear people say, well, genetic testing only measures a small amount of our DNA, that's where that comes from, because chip testing is really only pulling up to maybe a million. Most chips have anywhere from 600,000 to a million SNPs out of our DNA to find them. It's also the basis for ethnicity projections, where they look for certain SNPs that are present in some ethnicities more than others. They can compare your DNA and the SNPs you have against whatever reference library they have for ethnicities in order to tell which ones you're closest to. It's also how they do segment matching, because they look for the individual patterns of SNPs that you have that other people share as well, and they can determine shared segments from that. So in any case, they target those SNPs, they put them on a chip. You can then mass produce that chip and repeat the same test over and over again with different people's DNA. That's one of the reasons that autosomal testing is much cheaper than other types of testing, because these chips can be mass produced, and that's where most of the hard labor comes in, is creating the chip in the first place. Once you've done that, you can apply it over and over again in the testing process, and that can be done relatively cheaply on a lot of people's DNA. So the way it works is they put all of these receptors onto a particular chip called a microarray, and then the DNA for an individual tester is passed over those. Each of the little spots has a receptor on it that fluoresces, meaning it glows when those SNPs are found, and you identify the patterns of both positive and negative SNPs that way very quickly. And it's just as important to read a negative read in the SNP as it is a positive, because that tells you the SNP is either there or not. And if it's not there, that might point you to either a certain shared segment or an ethnicity, just as much as having the positive one too. So a couple of things about microarray testing, right? I mean, the chips that are created have a defined set of SNPs on them. The companies have some individual control over what that is, and they can look for things like certain SNPs off the Y chromosome. So that's why companies like either 23andMe or Family Tree DNA's Family Finder can use autosomal tests to report a very high-level Y DNA haplogroup, because they look for certain SNPs that will tell you what that tester is positive for, and they can find a high-level haplogroup from that. But it's not a discovery test, so they can't find down to your most recent haplogroup from those. But also the chip version matters. And the chips are compatible, the chip versions are compatible between themselves, so that you can look for the same SNPs on them. But the chip version matters as to, for instance, how deep on the Y chromosome you can go, or what SNPs you're looking for. So the ethnicity reference databases are either more or less detailed in certain cases, because they're looking for different SNPs that will help them with certain ethnicities or whatever. So the chip version matters. When you hear people ask what version a particular test was run on, it's because they're trying to drill down to the individual differences between those chips, whatever that may be for them, that's important as to what set of SNPs they were looking for. Now, you contrast microarray tests with our first detailed DNA test is called Sanger sequencing. So this is an original test. This was developed, like I said, first back in the 1970s, but it's very relevant today because it's very useful for reading short strands of DNA. So it's typically used, like I said, for individual STRs and SNPs. A lot of it is used on the Y chromosome. It doesn't have to be the Y. It could look at any chromosome you're looking for, but you're essentially pulling a segment out of the reference genome, in this case, and you're saying, I want to read that. So you create a primer for that desired range of DNA, and then the actual tester's DNA attaches there, and modified nucleotides are introduced. They bind from that primer forward until a fluorescent modified nucleotide joins that prevents further binding to attach there. The method is very useful because that section of DNA then is read off. It actually reads the complement, so the opposite, but you can recreate what the DNA strand would look like then if you know the complement. So then you have a read of that particular section of DNA that you wanted to pull. Obviously, though, you have to target a particular section of DNA. So you can look for unknown mutations. In fact, this finds them very often, but the section of DNA that you're pulling is very specific. So, for instance, this is not good for looking at parts of the reference genome. For a part of your DNA where you don't have a reference genome, there are still parts of our DNA that are not included in the reference genome. This can read them through other methods, but it's not good at targeting those. It's better if you can create a primer for a known section of DNA and then go look and see what mutations exist there. So again, it's very good if you want to target a particular section and look at whether there's a mutation there. Not so good if you're trying to read the entire whole genome of someone because it's time-consuming, expensive, and a little difficult to map together. Now, bear in mind, this is the original way that our entire genome was mapped back when it was first done, at a cost of something like $13 billion US. But that's not done today. We've moved on to next generation sequencing, which is the next thing that we'll talk about. So, next generation sequencing is the technology that underlies both whole genome sequencing and tests like the big Y700 today. This basically takes the idea of Sanger sequencing and multiplies it by, a million or 10,000. It's taking very short reads, so it's breaking our DNA into very small pieces and reading small sections. The reads are typically anywhere from 100 to 150 base pairs in length. We'll talk about what that means in a second because that does vary greatly between these types of testing, so we'll cover it on its own. But the same basic idea applies that the DNA is denatured, so the strands are split. It's fragmented up into little pieces. In this case, though, instead of reading an individual strand of DNA with a primer, you're binding these fragments on a flow cell, so you're getting a lot of them all at the same time, and then you're going through the entire flow cell reading individual nucleotides along each fragment until you get to your maximum read. And so then you get all these very short reads. They're put all together, and then you have software that aligns and merges the sequences back together in order to recreate your full genome, or the full section of DNA that you're looking for. Now, a couple of things about this. First of all, at the bottom, you can see this is intended to show your DNA and the number of reads for each part of your DNA. The reads will vary, so you'll get some parts of your DNA that are read over and over again and get lots of reads. Some parts of your DNA won't get read at all, or will only be read a few times, and you may not be able to tell what's there because you can't rely on each individual read. You have to put it all together and see if you get quality reads in aggregate. So with next generation sequencing, you're taking very small pieces and you're trying to put it all together. The end result is you do have a massive puzzle. Again, I go back to the analogy of trying to put a puzzle together. You've got millions of different little pieces here that you're trying to align after the read process is done, and that's mostly done with software, and it's a piecing algorithm that puts it together, usually against the reference genome. Again, the picture on your puzzle box that allows you to be able to get more quality in terms of putting it back together. But this is a whole software process. You get massive amounts of data, and then the software has to put it together. This is one reason why, after your test is run, it can take a while for next generation sequencing to produce the results, and they may have to be double checked because there's a lot of software that goes behind this in order to put it all back together. Often a human has to look that over and decide whether or not it called all of these pieces correctly or not, and maybe adjust a few things here and there. So you'll see some adjustments after the next generation sequencing test has been completed in order to clarify things that a human has looked at and said, no, that wasn't quite right. The software didn't get it quite right here, and put it back together. So that's one of the reasons why next generation sequencing can sometimes take a little longer, because it's got a lot of massive amounts of processing behind it. That's one of the reasons why you'll see it get corrected after the fact. So with all this data coming out of a next generation sequencing test, it's probably good to talk about some terminology that you'll hear about it. So here's a diagram. At the top you've got a reference genome, a sample reference genome. The actual DNA strand that you're trying to recreate is at the top as well. This is the actual tester's DNA, let's say. And in this case, there's two SNPs that are in this region of DNA. There's the actual user has an A instead of a T in the first place there, and a C instead of an A. So that's what you're trying to recreate, is that actual strand of DNA. You've got areas of the test that have produced reads in high coverage, no coverage, or maybe low coverage. Now, obviously, the amount of coverage is greater than in this example. That's where, when you see that a test is targeted at like 50x versus 60x versus 15x and so on, that's the read depth that it's targeted for. Now that will vary quite a bit across the whole genome. You'll get some areas with much more than that, and some areas with much less, and some areas with no coverage at all. So this is just a representative example that's been shortened down a bit. The read length typically is for what's called short read next generation testing, is usually between about 100 or 150 base pairs. It actually doesn't vary that much because you're reading that much of the fragment, trying to read it repeatedly. In this example, I've obviously shortened it down quite a bit too. But you've got an area of high coverage on the left here where you've got quite a number of reads that have consistently reported an A in that one position where the user has a SNP. There's one particular read that's been misaligned, and that happens from time to time. And then you have an area of no coverage. Sometimes that happens too. Even if a targeted test has tried to cover an area, sometimes there's nothing reported for various reasons. And then you've got one area of low coverage where you only have a couple of reads. And that's actually, in our example, has even expanded into an area where the reference genome doesn't cover. The reference genome doesn't cover 100% of our DNA yet. There are still gaps, particularly on the Y chromosome. There's an important gap. But even on other chromosomes, it's not 100% coverage. There's point something of gaps that still remain. And so sometimes you'll get reads over an area that the reference genome doesn't have a representative set of actual base pair values for. So in any case, when you hear people talk about read depth versus read length in a next generation sequencing test, that's what they're referring to. And the coverage can either be high, low, and that does depend on the company. Some companies call about 10 reads enough to call a SNP, and some will call with fewer reads than that. So that's not always consistent across the companies that are testing your DNA with next generation sequencing tests. Everybody has their own measure of quality there, and sometimes you'll get a report of a particular area that's reported with few reads. So you kind of have to take that as a representative call, but may not be entirely accurate. So you have to decide whether, if there's a SNP that's been reported there, if that's accurate or not, and that's on you to decide instead of the company. In any case, there have been some variations of next generation sequencing testing that have come out over the years that this has been available. Not all of these are available as products today. The two that are still available, of course, you've got whole genome sequencing, which is an attempt to do next generation sequencing across your entire genome. It reports as much as it can. This is not targeted, so it reports everything. But for genetic genealogy results, that translates into autosomal DNA, Y DNA, and mitochondrial DNA. At the moment, you get all this data back. There are no matching databases, so it doesn't identify matches. You have to translate the data into formats that can be uploaded to the current matching databases that are based on microarray testing. We've talked about that before. So you have to, this has to be translated through tools in order to get you a format that can be uploaded, and of course you lose a lot of the information because microarray tests don't pull every single nucleotide out of your genome. But this is the most comprehensive DNA testing that we can get today, is whole genome testing, and you can upload it to some of the sites that can do health analysis and so on. Then you have what's called targeted sequencing, which is basically next generation sequencing, where these flow cells are built to only target certain areas of your DNA. Particularly the one that's available today, the big Y700, targets the best regions of the Y chromosome that it can find in order to report SNPs in those areas. And that's where we get the heavy focus on Y DNA testing from family tree DNA, for instance, today. So that's called a targeted next generation sequencing test, very similar to whole genome, but it doesn't go after your entire genome. The two that I don't believe have actual products available today, there's one called de novo sequencing. Most of the whole genome sequencing and targeted sequencing build the profile of your DNA as compared to a particular reference genome. In the current environment, that's HG38. There's a lot of people that have been talking about the telomere to telomere reference genome that's available now, but that's a different subject. I'll leave that for now. But if you do de novo sequencing, that's essentially next generation sequencing, where you're piecing back the genome based on based on the puzzle pieces without the box top, if you will. The coverage quality depends on the read length, again, how much you're reading, how much of the fragments you're actually trying to read at one time. Longer is better because you can more easily build the actual puzzle back together with fewer pieces. But also it depends on the continuity of the sequence. The fewest gaps will make that more easy. So it's hard to do in places like the Y chromosome where you have large gaps. Then you have what's called linked read sequencing, which is standard short read, but they've tagged the fragments or the reads to better mark where they came from on the chromosome so they can recreate it more easily. It's a bit like if you tagged your puzzle pieces with where in the puzzle they appeared, so you could move all those pieces together and have an easier job putting the puzzle together. The only product that I'm aware of that used this was Full Genome's Chromium long read test, which is no longer available. It was $2,900 US when it was available, so it was relatively expensive. And I don't know if linked read sequencing drives that kind of cost increase, but in any case, there are no current products available today that use linked read sequencing. So now we've covered the three types of DNA testing that are available today that support all of the consumer products that support genetic genealogy. You've got autosomal testing and mitochondrial DNA testing that are done through microarrays, and chip testing, as it's called. There's Sanger sequencing that mostly supports single or panel STRs and SNP testing. And then you've got the commercial products like Big Y700 or the whole genome sequencing tests that are done with next generation sequencing, also known as short read next generation sequencing. Because the products that are coming down the pike and may be available, I'm going to say personally, let's say between 1 and 3 years. So they are being used regularly in genetics studies today, sometimes supported by next generation sequencing in order to prove their accuracy, but they're not quite ready in terms of accuracy and affordability to be provided to the consumer market for genetic genealogy yet. There are two types. They're mostly lumped together into the term long read testing or long read sequencing. One is from a company called PacBio. It's single molecule real time. It's called SMRT. Also, it is also branded under the name HiFi. And then you've got Oxford Nanopore Technologies Nanopore test. But if we look first at the single molecule real time testing, you've got a very similar concept to flow cells, but in this case they're small chambers that are called zero mode waveguides that are filled with fluorescent nucleotide dyes, and the DNA strands are pulled through those, and as they pass through a small chamber at the bottom, the dyes will bind to the DNA strand, fluoresce in the light that's shown through, and that then is read so that as each nucleotide passes through that tiny aperture, you can illuminate it essentially, and with the help of the fluorescence, you can read what the value is. So that's single molecule real-time testing. It is able to pull longer strands of DNA than what we typically call short read. And then with Nanopore, it's a similar concept where you're pulling a longer DNA strand through an individual part of a bigger flow cell. But in this case, you've got a small pore of nanometer size that you're pulling it through, and there's electric currents in the pore that will measure the individual nucleotides of that DNA strand. So it's the same concept of pulling it individually through one part of your cell, but in this case you're measuring it with electrical variations. Now, I've said over and over again that these new technologies give us long read capabilities as opposed to short read. So why does the read length matter? Again, when we go back to look at the technologies that are based on reads today, you've got Sanger sequencing that typically is between 500 to 600 base pairs. It can be up to about 900 or so. It does vary in the length of reads that it can use. You've got next generation sequencing, which is typically between 100 to 150 base pairs. It can be up to 400. I don't know of any tests currently available that do that, but that's what drives the label short read on those, right? So because you're breaking your DNA into very small sections that you have to put back together in alignment. The third generation sequencing, what I just talked about, Nanopore or SMRT, can do much larger strands of DNA. So between 10,000 and 30,000 base pairs is common. It can do up to 2 million. I've seen recently PacBio is advertising 1 million base pair reads, and actually they say they're using linked read sequencing. I don't know if it's exactly the same as what I just explained, but presumably it's close to that. So they can do much larger sections of DNA at a time. Why is that important? Well, longer reads yield more reliable alignment because you're working on basically bigger puzzle pieces. So it's the equivalent of going from a 1,000,000-piece puzzle down to a 1,000-piece puzzle or something like that. So that allows the software, after the test has been performed, to map the larger DNA structures better and to more reliably put your DNA back together again with fewer gaps. So you can get larger STRs out of it, you can get structural variants, you can map some of the larger mutations that may affect our genes, so it has implications for health data, but also for genetic genealogy. We might be able to use more of the mutations in our chromosomes and also get more mapping of our chromosomes as well, especially in places like the Y, where some sections of it are not easily readable through short read DNA today. And in fact, just using the Y chromosome as an example, it has a large section called the Q12, which is a region of highly repetitive DNA. I've illustrated it here just with some example sequencing of longer strands of repetitious DNA rather than the rest of the chromosome that has fewer of those sequencing, not entirely free of them, but fewer of them. And what happens with short read next generation sequencing is that it's easier to map a short read to an area that's less repetitive because you can more easily identify where it came from. With an area that is more repetitive or subject to more confusion, smaller pieces of the puzzle can map to different places on the chromosome, and it becomes more difficult to know if you're actually reading the section of DNA that you're targeting. With longer read, that problem doesn't go completely away. You may have one or two of these reads that don't map well to a particular area, but the rest of it maps just as well as the rest of the chromosome, so you can cover those more repetitive regions with more quality, and you can align better in areas where you can't align as well with short read next generation sequencing. So that's one of the reasons that long read has the potential to give us an improved DNA testing capability that we don't have today. Now, I don't want to minimize short read next generation sequencing. The advent of NGS has given us, since about 2013, an explosion in SNPs, an explosion in testing capability, and in the capability of matching ourselves against each other that has done wonders for Y-DNA testing and for DNA testing in general. But we do have the capability to get another leap up in quality improvement from long read, and we'll have to see if that pans out. Okay, so that completes the circle. I've talked about all of the types of DNA testing that we either have today or that should be coming out on the market soon. Let's talk for a little bit about the types of files that you get currently from next generation sequencing, but the prediction is that we'll get something very similar from the long read technologies as soon as those products are available on the market. So when I talk about the files that you get out of these DNA tests, I typically make an analogy to an X-ray that you're getting at the doctor's office. It's not a perfect analogy, and certainly the folks who understand the DNA testing better would argue with me about some of this, but it helps, I think, to try to understand why you get so many different versions of files out and how they can be built on top of each other and translate the scientific data from before into something more easily understandable to those of us who do genetic genealogy. Most of the next generation sequencing tests today, at the raw data, they'll produce something called a FASTA file or a FASTQ file. There are some differences there, but don't worry about that right now. This produces the raw sequencing reads, and it's also got the read quality score, so how well did it actually read the fragment itself. This to me is analogous to the raw data. If you go into the doctors, you need an X-ray, they take you into the room, they do an X-ray. This is the raw data that comes out of the X-ray machine even before the picture's been produced. The picture, in my analogy, is more analogous to what's called the BAM files. Now again, there's different versions of this. A BAM file is typically a binary file. There's SAM files, there's CBAM, there's UBAM, that's unaligned BAM files, and so on. There's different versions of a BAM file, but the basic idea there is that is the reads mapped to a particular reference genome. In today's world, that's mostly the HG38 genome, but it has enough information there in order to map it to any other reference that you want to, which is why some people will submit their BAM files and have it remapped today to, let's say, T to T, which is a different reference, and your BAM file can be used to produce that data as well. That just translates your BAM file, if you will, into a different language in some way of looking at it. To me, this is analogous to the X-ray picture. It's very useful. You can port it around. You can give it to another doctor. They can read it. They can tell you what that means, so it's easily interpreted. The BAM file typically has both read and mapping quality scores, so that's a different quality score that says how well the reads were aligned and mapped to particular sections of the reference genome. Those then get translated into something called VCF files, which typically is the first time that you'll see variants mentioned. The BAM file doesn't say what SNPs you have. The VCF file will. So this is where they've interpreted those reads into variants. It may also include some summary quality information, but it typically doesn't include everything that the BAM file does, for instance. To me, this is analogous to the doctor's notes out of the X-ray. It's their translation of what the X-ray means. So for you, it may be enough data to get everything you need out of it, but it's not the X-ray picture itself. If you took it to another doctor and said, give me your second opinion, they'd have to interpret the doctor's notes and say, well, I can't see the X-ray. I don't know if they were right or not, but here's my interpretation of what the doctor saw and said based on what he or she had in their notes, not based on the X-ray picture itself. So you often want the X-ray picture for certain things, but sometimes the notes are enough. Sometimes the VCF file is enough. Now, every company produces other types of files that you can use that give you the results. Those are typically output from their own internal databases. They're not identical to VCF files. They're not really, they are sometimes a portion of what's in a VCF file, so they're not always translated directly from VCF files. But these include files that may have the suffix of .csv, .out, .tab, whatever. All of these will give you summary information about your test that may include SNPs. It may include a lot of different information, again, that's often also in the VCF file, in formats that are easier for you to read or to analyze, but they don't give you as much information typically, certainly not as the BAM file, but often not even as much as the VCF file. And I tend to use the analogy that this is analogous to the summary information that's entered into a patient database. It may not even contain everything that was in the doctor's notes. It may just be typing that in as well, so you might get the same information there. But that's highly variable as to whether or not you have enough information there if you want to analyze your complete DNA test and find things that, in this analogy, the doctor didn't find. You really need to go back to at least the VCF file, if not the BAM file, and find out the original information. You don't generally have to go back to the FASTA or FASTQ files to get that information. The BAM file is usually enough, though. So when you see the different output that's produced by the company based on your test, there are different levels of interpretation that have been applied on your data, and you want to know which one you're targeting if you want it for a specific. Right. Now that may be all you ever wanted to know about DNA test types that relate to genetic genealogy. If it didn't, if it's just whet your appetite to learn more, there's a number of places that I would suggest you start with kind of as the next level down. Randy Harr actually has a wonderful website that he's put up that has a slew of documents and articles that he's written on various aspects of next generation sequencing testing mostly. He focuses on whole genome sequencing, but a lot of what he writes is also applicable to the more targeted NGS tests like Big Y700. And his link is here on the left-hand side if you want to go there and get a lot of information about read depth, read length, and a whole slew of other aspects about whole genome sequencing. There are also explanation videos that go kind of a next level down. I've split them out here on the right: Sanger sequencing, next generation sequencing in general, SMRT, HiFi, as well as Nanopore. If you want to go to any of those, you'll have to copy these individual links from here, or I'll try to put them all in the description of the video as well online. So if you want any of them, you can just go there and hopefully click on them and get to the one you're interested in. I hope that gives you a lot more information to explore on these, and thank you.